Hi, everyone. Thanks for joining. We're going to wait just a minute for folks to trickle in, and then we'll do some quick announcements and get started. Okay, so it looks like the trickling is slowed down, so we could go ahead and get started. So we have two great speakers today, Rachel Poretsky from UIC at the University of Illinois, Chicago. I think there's a little typo there, Seattle, it's probably my fault, sorry about that. And Irene Xagoraraki, sorry if I mispronounced that, Irene, at Michigan State. This is our last seminar for the summer, so for the next 2 months we won't have them, and we'll take a little summer break, and we'll send an announcement when we restart essentially with the semester in the fall. A reminder, we have a workshop coming up on July 13th. The info's here. I'll wait just a second if you want to grab a snapshot with your phone. And I'll after I get done with the announcements, I'll drop the link in the chat as well if you'd be interested in self-nominating or nominating someone to participate. And that's on the ethics of wastewater surveillance for infectious disease. And we just finalized the date for our annual meeting. It'll be August 2nd. It will be a 24-hour period with virtual poster sessions and panels, but there'll be a live portion from 12 to 2 Eastern time on that day, and that's Wednesday. So we'll send out more information shortly, but essentially anyone who's interested will be able to present virtual posters, and it's going to be a little bit of a unique format. We think that will be hopefully really exciting and allow a lot of participation. So with that, I will stop sharing my screen and allow Rachel to get started.
Super. All right. I feel like it's tradition. I have to ask if everybody can see my screen, but I'm pretty sure it's all good. Excellent. So I'm joined today by a PhD student on our team who's going to talk a little bit at the end, Christopher Owen. And I'm going to talk today about less of a targeted approach and more of a sequence-based broad surveillance approach to looking for targets in wastewater. And when Ali asked me to talk, I knew that we had data coming. And so I asked for a very, the last date so that we could share some new data. And we have some brand new data, but not all of it. So we'll be able to share some of that. But before I get into things, I just want to share a little bit about our team and our people. And this is not even all of our people, but we have this huge collaboration between folks at UIC and Argonne National Lab and Northwestern University, who are kind of linked together through the Discovery Partners Institute, which is a division of the University of Illinois system. And so on the top, we have some of our key leadership folks, and on the bottom, we have some of our key lab folks who work here at UIC in my lab. The first 4 people are lab managers and technicians, and Leah, the second to last, is a master's student who's graduating in a couple of weeks, and Christopher is sitting right next to me. And we also have partnerships, broad partnerships across the city and across the state with the local public health departments. We were initially funded by the Walder Foundation, which is a philanthropic philanthropic organization that funded us in 2020 when we got started. We worked together with lots of commercial partners and local partners to help us both manage our samples, manage our data. And here's a fun picture that's a little bit blurry, but our team won a Chicago Innovation Award in November, which was great fun. We got to dress up and be fancy and be acknowledged for this wastewater work. So a little bit about our program. So we started in 2020 just in the city of Chicago with a couple of sites. And in 2021, we expanded statewide with the idea that we would have location in each of the about 102 counties across the state of Illinois. And we're pretty close to that. We can't get all counties. It turns out we have many counties on septic, as many states do, but we are in actually 8 now, actually 11 sewer sites around the city, and some facilities including Cook County Jail, O'Hare Airport, some hospitals, congregate living facilities. We have a project funded with CDC to look at CORIS in hospital settings. And then the major wastewater treatment plants that serve the city of Chicago and the near suburbs. And then across the state of Illinois, it fluctuates, but we're about 80 sites. We gain some and lose some still on a pretty regular basis across the entire state. And we recently completed a pilot program in K-12 schools. So we're doing a lot across the city and state. And we do have a dashboard for our Illinois program, and like everybody else, or most people, we report to news, so we're up there too. And we've been doing a tremendous amount of sequencing. And so this is a summary from 2022, I think, to last week, are tracking variants across the state in the wastewater. So this is aggregated from the whole state, but we sequence over 100 samples a week, and we do whole genome sequencing for SARS-CoV-2. And I think, I don't want to say this without any backing it up, but I think we generate among some of the most sequenced data in the country for wastewater programs. So since 2021, we're pretty close. Maybe next week we'll get to our to 9,000 samples that we've sequenced. And so in thinking about sequencing, I kind of went back and looking at wastewater, I kind of went back to some of these projects that we did years ago. And so there, several years ago, I had a project with George Wells, who's in the middle, he's at Northwestern, and Brian Murphy is here at UIC. Dorothy Wright-Folks is now, she was an undergraduate at the time working on this project, and she is now our liaison to at the Chicago Department of Public Health, which is super fun. And Ben was a postdoc and Morgan was a PhD student at the time. But this project was to look at antimicrobial resistance in wastewater and in receiving water bodies that received wastewater effluent. And so we did a lot of sequencing for this project, and we did a more shotgun-based approach. And we really tracked antimicrobial resistance genes from influent through the wastewater treatment plants to the effluent. And we did this in different types of wastewater facilities. And one of the things that we were able to do is see total communities. So we got a sense early on in our time here, we got a sense of how complex wastewater communities are. And on the left is bacterial community, on the right is viral communities, and the viral, this is just DNA viruses too. So just giving a sense of like, the diversity and the overall microbial load in wastewater in general. And so when we were thinking about using shotgun sequencing to look for antimicrobial resistance genes, we actually used digital droplet PCR to quantify a number of antimicrobial resistance genes, and then calculate how much sequencing we would need in order to capture any of those in a shotgun sequencing data set. And so we, as an example here, I put up at the top, we would need it for SOL1, a sulfonamide resistance gene. In order to detect a single copy of that gene, we would have to get 10 gigs of sequence data. And we were able to sequence fairly deeply and see a number of resistance genes that's shown on this heat map here, and we were able to make overall observations about how many antimicrobial resistance genes there were and who they belonged to, who they were associated with, were they associated with mobile genetic elements and such. And so this really got us thinking, as we have been doing this wastewater surveillance work since the pandemic started, about how much we would need to sequence to capture some of these pathogens that we're interested in monitoring in wastewater. And, this comes from the idea that as, initially we were using the same primer sets as most of you on here, looking at, we're looking for SARS-CoV-2 in wastewater, and then different variants would hit, and we would scramble to generate new primer probe sets to quantify the new emerging, the gamma, and then Omicron when it first hit, and we were frantically trying to make BA1 and BA2 primers. And then there were Mpox last summer. And so we, like many of you here, scrambled to get Mpox primer probe sets and get those assays up and running. But the thing is that all of these targets are in the wastewater already. And so for us to generate primers for each new thing that we want to detect is a lot of work. And now we have GT Molecular, who we partner with too, who's doing this for CDC and generating sets for all the news targets, the things that they're looking for. But until last summer, Mpox wasn't on that list. So who knows like what might get added to a list, and then we have to develop new primers and probes. And so we were thinking that we could use a sequence-based approach to try to get at what's in the communities. And so I think I'll turn it over to Christopher, because he's leading this work, and I thought it'd be nice to hear it from the lead himself instead of me.
All right, so we did a couple of sequencing runs. Initially, we just sequenced 3 samples and sequenced them very deeply. This figure on the right shows dPCR results for SARS-CoV-2 targets. The 3 purple stars represent the samples that we selected, so they had particularly high SARS-CoV-2 concentrations. We initially just used the same concentration and extraction protocol that we've been using for dPCR, which involves concentrating samples with nanotrap microbiome A particles. And we also did one of the samples with a couple of different methods, including one not concentrated. So we just extracted nucleic acid directly from the sample, and one that was concentrated with an ANOVA prep concentration pipette. Uh-oh.
Click again.
Okay.
Sorry.
So these are the results from those initial samples at a very high taxonomic level. We were surprised that a lot of the sequences weren't classifiable based on using a Kraken2 classifier against a database that includes from RefSeq all of the bacteria, archaea, viruses, fungi, and human. So that's the largest fraction for the DNA, is just couldn't be classified at any level, and for some samples with the RNA as well. Other than that, vast majority of the sequence data came from bacteria. We were largely interested in viruses. There is some interesting data within the bacteria, but most of what we're interested in is viruses. So I'll go more into what we're trying to do about that in a minute. But since we sequenced the samples so deeply, we were still able to see some virus data in them. The viruses for all of these samples accounted for less than 1% of sequence data, but out of a few 100 million reads per sample, that's still some amount of data. It also happened that most of the viruses were from non-human infecting taxa. So for RNA, the most abundant virus by far was from phylum Chytrinoguricota, which is a phylum of plant viruses that includes PMMOV, which I'm sure a lot of you use as a fecal load indicator. And in DNA, again, a vast majority this time was from phylum Uroguricota, which is a bacteriophage phylum. It includes the crossphage. There's a tiny bit of other stuff. I don't know if you can see it at all, but at the top of the pink bars, you can see all of the other taxa. If the resolution is coming through high enough, you might be able to. And then just filtering out the family Coronaviridae to look at coronaviruses. There are not very many. And again, this is from the samples that were selected because they had particularly high SARS-CoV-2 concentrations based on digital PCR. These samples are spiked with bovine coronavirus, which is a recovery control for PCR assays. So a lot of what's in this beta coronavirus 1 taxa is bovine coronavirus. The severe acute respiratory syndrome related coronavirus, since this is visualized at a species level and SARS-CoV-2 is a subspecies, it doesn't show that tax ID. But all of the reads within this species were SARS-CoV-2. They only showed up in the sample that had the highest concentration of SARS-CoV-2 and one of the highest concentrations that we had measured for the whole year. And this only amounted to about 20 reads that were identified as SARS-CoV-2 out of 400 million or so in this sample. It's maybe worth noting there could be other reads that came from SARS-CoV-2, but they matched other taxa equally well. In that case, they might have been assigned to one of the higher taxa based on the classification method we were using. But the only thing we know for sure that SARS-CoV-2 is extremely low abundance. So in an effort to get more viruses, we're trying a couple of different things. One is using sequence targeted panels. So there are a few companies that make these. They're designed to enrich specific sequences from a sample. We also used some size selection. So that's what's shown in this picture. Samples were filtered through a 24-well plate. that contains filters with a pore size of 0.4 microns, which we're hoping would remove a lot of the bacteria, but not all of them, because there are some bacteria smaller than that. So we've done some sequencing with one of those sequence enrichment panels, the adventitious agent panel, which is made by QIAGEN. We have some preliminary data from those panels. This is from a sequence depth of around a couple 100,000 reads per sample. So these are numbers of sequences that matched some target within the panel. So these were classified against a database that is provided by QIAGEN that includes all of the taxa in their adventitious agent panel. So we still actually see relatively few reads from the targets that were enriched. It's not shown here, but most of the reads were still bacteria, even though we didn't enrich for them. And this is another way of showing the same data, just a different reclassification method. And this is all of the samples combined. So the majority of hits are adenovirus or astroviruses. That's not to say that it's the majority of sequences in the sample, but it's the majority of hits to the database of enriched sequences. And we also processed the samples in a couple of different ways. Yeah. Let's see, I think this.
Can we move this?
I don't know.
The Zoom presentation is blocking the legend at the top. It's okay. It's
Okay.
divided into different concentration methods.
You can see what the concentration methods are at the top. So we sequenced, a lot of them are the same samples with different concentration methods. Not every sample was sequenced with every concentration method, but a lot of them are the same. This is very new data that we've just started looking at this morning. But it looks like maybe there is some difference between the concentration methods, especially, I think the one at the left was direct extraction without any concentration. Yeah.
I can just finish up that we have more data coming, and I think what we're trying is a balance of panels and shotgun sequencing to try to get at some of the lower abundance targets in our samples in unbiased or less biased way. So we can end there, and with that, take any questions.
Great, thank you. And just a reminder, I forgot to mention this at the beginning. Any questions you have, please put them in the Q&A box, and you can also upvote different questions once they're in the Q&A box for the speakers. So the first question we have from Mark Johnson: have you tried doing an assembly on your unknown sequences, or what have you? I'll add on to that. What other approaches have you had to try and annotate or look into those a little bit more?
Yeah, do you want to take that?
Yeah, we have tried doing an assembly. It is something that I spent not a ton of time on a long time ago. If I remember correctly, I think a lot of the assembled contigs were still not identifiable. Didn't map to anything in the database. And the taxonomic breakdown looked pretty similar based on reads that mapped to the contigs and then with those contig IDs. Yeah.
Yeah, it was an initial initial analysis to try to get at the unknowns, but yeah, it's a good question.
Yeah, I mean, following up on that, there was another question. Do you think some of your unknown sequences could be yeast? So have you all explored other, again, adding on to that, have you explored other databases to compare against, or are you pretty much solely focused on the viral fraction?
Oh no, they're in the database. We use a whole all domains. So in fact, Christopher didn't mention it, but some of a small percentage of the reads were human, a small percentage. I think most of the eukaryotic reads were human, right?
Yeah.
Yeah. So there were some.
A good chunk of the eukaryotic reads that weren't human were yeast.
Yeah.
Great. Another question from Rose Cantor. What was the depth of the deepest sequencing run that you did? How deep did you sequence?
About 400 million reads per sample.
And the only one that had SARS-CoV-2 in it was the highest COVID levels, community COVID levels.
Got it.
Yeah.
The next question is, I'm curious about your wastewater sampling at the Cook County Jail. What is that process like, and are the results communicated to the community or the facility?
Yeah, so we work closely with the sheriff's department and the health. So there's the sheriff department that kind of runs the jail, and then Cermak Health does the medical stuff at the jail. Sorry for stuff, but yeah, so we work with them with the sheriff's department to sample, and we work with the infectious disease team at the jail to report our data. We report weekly, and it's a fantastic relationship. Like, he'll respond with the ID doc will respond with, the wastewater either matches what they're seeing in cases. We do influenza, RSV, SARS-CoV-2. We do a bunch of things, and so he'll let us know whether it matches what he's seeing in clinical cases at the jail, or he'll say, oh, last week's data was told us that it was going to get high, and now we see it, we found it. So it's really great. And we also report to the Chicago Department of Public Health, so they see the data as well. And we're piloting, this is a whole separate thing, but we're piloting, we're doing a pilot project also with the Rush University and the Center for Emerging Infectious Diseases to look at air sampling and wastewater. So in the intake unit of the jail, we've had for about 3 months an air sampler running, and we're doing a qPCR panel that gets 8 different respiratory pathogens, bacteria and viruses, and we're comparing that to the wastewater sample that we get from that unit. So we get from a number of specific divisions within the jail. Yeah, it's fun. It's a cool project.
Very cool. Another question from Lauren Stadler. How do you envision the integration of the sequencing and targeted digital PCR approaches? And then following up on that, do you have a sense of how quantitative the sequencing and enrichment panels are?
Yeah, I don't think it's going to be super quantitative. It's going to be relative abundances, but I think more what the strength will be in either detecting things that we weren't targeting with a PCR assay, or seeing relative abundances shift. So we can even do things like completely agnostic, where we're looking at getting shotgun sequence data and looking at like Kmers for an example, and looking for Kmer patterns that might be increasing exponentially, and then try to figure out what those are if that's increasing. But I don't think it's going to be super quantitative. It's going to be more like more trends and more relative abundance. Would you agree?
Yeah.
Thank you. Yeah. So one last question, and then I'm sure there's a few more I wasn't able to get to in here, but I'm sure that the speakers would be glad to reply in the Q&A box.
Yep.
It's on the different concentration methods. Do you have thoughts on what is driving those shifts between the methods that you're seeing in between the different concentration methods you tried?
I wish we knew better. I think that we're hoping to get at that with a whole bunch of sequencing, different sequencing approaches on the same concentration methods and then on different concentration methods. But part of the issue and part of what the impetus for doing that first deeply sequenced trial was that we were using the nanotrap particles and that were supposed to, initially they were called viral particles, now they're microbiome particles, but they were supposed to enrich. And so we thought maybe if we're already enriching for viruses and we sequence that, maybe we don't have to do anything else. And it turns out that they're enriching for lots of things. We don't really understand how they work and what the differences between the different nanobeads are. And so the short answer is we don't know, but we're hoping to try to figure it out a little bit.
Yeah.
Great, thanks. And like I mentioned, there was a few I didn't get to, so I think we can respond in the
take a look.
Q&A. So with that, Irene, you can take it away.
All right. Thank you guys for a great presentation. Very interesting work. So thank you. I really enjoyed your presentation. So let's go to Detroit here. What are we doing in our region? All right. So first of all, I'd like to introduce the project partnership. We work with the Great Lakes Water Authority, the City of Detroit, Water Department, CDM Smith, Wayne County, Macomb County, and Oakland County. And here are a few pictures of us sampling at the wastewater utility and also at in the sewer sheds out in the streets. And also a couple of pictures of us in the lab. Now, I'd like to spend the second explaining the project evolution in Detroit. We started in 2017, prior to the pandemic, with a National Science Foundation grant looking at wastewater-based epidemiology. Then CDM Smith gave us a grant to transition to COVID, and at the same time, the Great Lakes Water Authority funded us to switch to COVID SARS surveillance. Then we got funded by the Michigan Department of Environment, Great Lakes, and Energy, and then the Michigan Department of Health and Human Services. So we are currently funded by the Michigan Department of Health and Human Services and by the Great Lakes Water Authority, and our current grants are for the next year. So I'd like to start with some examples of our work, starting from the beginning, 2017, because at the time, there was no COVID, so we were looking for other pathogens. So I'll start with that, and then I'm going to skip the COVID work. I'll just very briefly describe it, and then I'll move on to other pathogens and our approaches that we're doing now. Okay, so when we started up this work in Detroit, there was a hepatitis outbreak. So there are, we worked with hepatitis A. This is an example of a published paper that we did. If we look at the clinical data here, we see that we had a peak in clinical cases in Michigan, in our region. We did do some sequencing, and we were able to identify in 2017 and 18, we were able to identify hepatitis A, B, C, and D. And then we focused on hepatitis A. So the blue dots show confirmed cases during the pandemic in 2017 and 18. In the left graph, the right graph shows the later after the pandemic, after the epidemic, after the outbreak of hepatitis A, so all the cases dropped down. And at the same time, we were measuring hepatitis A in wastewater, and we see that that we were able to capture the trends. And actually, if you look at the dates, we plotted the concentrations of wastewater like 2 weeks ahead of time. So we were able to predict the peaks about, we had a lead. So we were able to predict 2 weeks before the actual peak. So that was quite interesting, and it showed the power of wastewater-based epidemiology. Then we thought of doing a sequencing study to look see if we can see any other viruses in the sample base, like something like the previous presentation that we just listened. So this work is published, and we did do Illumina sequencing, and as we saw in the previous presentation, the majority of sequences are unidentified, and we have some bacteria sequences, but we were able to capture about 3% of our sequences were viruses. We did a virus, we used the Viradel method for, which is an EPA method approved for isolating viruses from water samples. And then we saw a lot of bacteriophage, but in this graph, I'm focusing on the genuses that are related to human viruses. So we were able to see blood-borne, vector-borne, enteric, respiratory viruses in the samples. That's before the pandemic. So we saw some coronaviruses at the time, but they were alpha coronaviruses, like the common cold. We saw a lot of orthopox virus, which is related to vaccinia, the vaccine strain. And the method is good for testing virus genuses. If you want to go to this species, the strain level, so is it like SARS-CoV-2 or is it like a beta coronavirus, then you gotta do additional sequencing. But this works well as a screening method, and I'll talk more about that regarding the work we're doing now. And then we also did some work on herpes viruses. We were able to, this is with PCR, we were able to detect, 8 strains of, oh man, I can't control my slides. We were able to see 8 strains, and we saw that herpes virus 8, which is quite uncommon in the United States, was very abundant in Detroit. So that raised some concerns, and we did some sequencing. We saw very similar trends. And then we were able to relate that surge in herpes virus 8, which is related to Kaposi sarcoma. It was we were able to relate that with an outbreak of HIV that was happening in Detroit at the time. So immunocompromised people are more prone to herpes virus 8 infections, and this was all done with sequencing. And then COVID came, and we switched to SARS. So, but this presentation is not focusing on COVID. So I'll just briefly say that we were able to publish our work. We were able to have like 5-week warning for COVID peaks in Detroit prior to Omicron. We did some involved time series methods. We also published another paper with simple methods that can be even used by the public, like when we upload our data on a public database. So when you look at the daily numbers, then you can kind of use those simple methods to see what's coming. And then we also did surveillance at the neighborhood side, and we were able to assess per capita estimations, and we looked at some biomarkers. But moving back to the topic of today, which is surveillance beyond COVID-19, what we did is this paper is funny, but it appeared online today. It's the first day that it's available online at the Frontiers of Public Health. We developed a ranking system, wastewater surveillance before COVID-19, a ranking system for communicable diseases. So there's like 96 diseases that are reported by the state of Michigan. So if we wanted, none of us has unlimited funding and unlimited resources. So if you want to focus on one pathogen, how are you going to decide which one you're going to look at? So we developed this method, this system, CDWS rank, we called it, and we use 6 binary parameters and 6 quantitative parameters. The binary parameters are inclusion into lists or not. Like, is that disease part of bioterrorism list or so? I'll show that in a minute. And then the quantitative parameters, 5 of them are driven by clinical data, and 1 is the R0 value, which shows how contagious the pathogen is. And this is the our system here, where we have the binary parameters are presence or not is in the CDC National Notifiable Infectious List, presence or not in the data reported in the list of the reportable diseases in Michigan. Is it presence or not in the EPA contaminant candidate list, and presence or not in the bioterrorism agent list? And then the quantitative parameters, again, 1 is R0, which is kind of shows the infectivity of the pathogen. And then the others depend on clinical data. So we compiled clinical data between 2014 and 2021, and we looked at the trends with linear regressions, and then the geographic ratio, whether we're focusing on the city of Detroit, Wayne, Macomb, and Oakland counties. So we wanted to see if it's which diseases are more prevalent in our area. So again, we looked at clinical data. Some examples here. The red lines show the overall trends in Michigan, and the black lines show the trends in Detroit. So there are some diseases that are more prevalent in our area. For example, STDs are more prevalent in Detroit. GI, gastroenteritis, is more prevalent in the rural areas. So if we were doing monitoring in Detroit, we have to take that into account. We'll focus on the diseases that are more relevant to us. And then we also saw some trends in diseases affected by the pandemic, but that's like influenza dropping, Lyme disease going up because people spending time outside. But that's beyond the scope. I just showed that. And then this is our rank. These are our ranking scores for our prioritization scheme for the city of Detroit, the tri-county Detroit. Now this ranking is different for rural areas. So we see here that SARS came up top, of course. We have some STDs here. Some diseases like polio, for example, which we're doing now, even though there isn't any cases right now, but because of the R0 value that's high, kind of came up. So we see this as a guidance for the public health departments to decide which targets they're interested in and which targets we're going to focus on with a limited amount of funding. So for example, now we added polio and we're adding STDs. Speaking of that, I was at the conference very recently in Michigan, and someone from CDC talked, and he gave us an okay to take a picture of his slide. So I assure you that it's okay that I took that picture. And this is these are some agents that are prioritized by CDC. And some of them showed up in our list, in our prioritization list also. So people are thinking about new targets. So the last thing I want to mention here is our current sequencing approach, similar to what we heard in the previous presentation. Again, with us, we tested different methods. We tested the polyethylene glycol concentration, and we tested the Viradel, which is the based on positive electrostatic attraction, positively charged filter. We saw that with the Viradel method, we had about 3% of our sample was viral, and with the PG about 1%. So methods, pre-treatment methods play a huge role. Again, we were able to identify respiratory, bloodborne, foodborne, vector-borne, and other viruses at the genus level. So we were able to see beta coronavirus, right? But then we couldn't, with this method, I see this as a screening method. So what we're doing in Detroit now is running this once a month. And like, let's say if we see something coming up, like for example, lentivirus, it's related to HIV. We know there is an HIV endemic population there. If we use this as like how we use at the clinical world, if you take like a scan, like an ultrasound, and then or like a breast exam, like they take it every year and then they compare it with the year before, right? Are there any changes? I know that my doctor is doing that, right? So they look at the previous scan and this year's scan. Is there any change next year? Is there any change? So we're planning to do that every month and see if something pops up. If something pops up, then we're going to do targeted sequencing and targeted qPCR. So that's it. So this is our screening method together with the prioritization scheme that I showed before. And again, here we see the two different methods, and this is our bioinformatics workflow. My postdoc, Yabing Li, is an expert in that. And we did some streaming, annotation, assembly, etc. I think my time is up. So I will stop and I will be happy to take your questions.
Great. Thank you, Irene. And just a reminder for folks to enter your questions in the Q&A and upvote any questions that you're interested in. So I'll kick things off. I was really interested in the ranking priority system for developing other targets. You know, it seems like we're kind of picking them off, all these apples off the tree, and maybe it makes sense to think about it a little bit more carefully. It seemed like you primarily considered the disease importance. I'm wondering if you considered the suitability of these targets for wastewater monitoring. For example,
yes, we did.
Shedding or disease prevalence.
Great question. I forgot to mention we did. So overall here, we have each parameter is there is a weighing factor, and then we have a parameter here, pathogen detectable in wastewater or human excrement. So if it's not, then we multiply by 0. So we start with the 96 diseases, and then we did an extensive literature review. Are those agents detectable in either wastewater or human excrement? If not, multiply by 0, they're gone. If yes, then they stay in the system. So that's a parameter. And then an additional parameter we added is the disease associated with a single pathogen or a multiple pathogen? So for example, gastroenteritis, there are many pathogens that can cause gastroenteritis, but because there are multiple, it's not possible to test for all. So when we have multiple diseases, then we multiply by 0. Thanks for your question. I forgot to mention.
Yeah, thank you. And Cara Nelson had a question here following up on that. For this ranking system, is there a criterion related to how effective current clinical surveillance is?
Oh, no, we didn't. How effective clinical? No, we didn't have that criterion. That would be a great criterion to add.
Great. So I was also really interested. Oh, sorry, one just popped in here. What testing are you focusing on for the future?
Okay, so we keep our SARS. We are doing polio. We are doing the sequencing that I mentioned once a month. And we're thinking of some specific diseases for Detroit, which are STDs. And that's what we're thinking for right now. I wish it's all about money, you know.
Yeah.
You just can't test for everything. We are meeting every week with our health departments. We have excuse me, every other week, and we are talking. And if something changes, then we're going to change priorities. But now our additional one is polio and the STDs.
Great. Well, I'll ask one last question I'm interested in, unless we get another one in the chat, and maybe give folks a few minutes of their day back. So I was really interested in the herpes virus and some of these other viruses that you did early on before COVID. And especially something like that would be more of a latent long-term infection. I'm wondering what you think the relationship might be between those and cases where shedding might happen over a longer period than an acute infection like COVID, if you've had the opportunity to think about that relationship at all.
Well, what the literature shows actually is that those herpes virus infections are increased now with the pandemic, because when people are when you get another disease and your immune system is suppressed, then the existing latent infections, they get worse. I'm not a medical doctor, but so that question is relevant to whether you want to see what's happening in the population right now, or when you want to see changes over time. So I'm not sure maybe I didn't understand the question.
I'm basically asking, so for COVID, we've shown time and time again, there's a relationship between disease prevalence in the community and what we observe in wastewater. And I'm wondering if that relationship would be the same for something like herpes, it's a latent infection. Yeah.
The thing is that with herpes, we don't measure all we don't have clinical data for all species. Right. So we're going to have clinical data for a few. But I do think, based on our work, I do think that there's a correlation. Yes.
Very interesting. Well, thank you again, Irene. I'm sure if folks have more questions, she'd be glad to follow up over email.
Yes, and please. This is my email.
Yeah, and thank you everyone for attending today. Again, a reminder, we won't have seminars for the next 2 months, sort of a summer break, but we do have a workshop in July and our annual meeting August 2nd, which is a Wednesday. You'll see coming through the listserv. Thanks everyone again, and we'll see you at another event soon.
Thank you.
Thanks, Irene. That was great.
Thanks, guys.
Thank you all. Have a great weekend.
Thank you, Rachel. See you, Hessa. See you, Amanda.
Bye.